Malaria is an infectious disease caused by a parasite called Plasmodium. It is spread from person to person by infected Anopheles mosquitoes. The parasite's life cycle is complex. When an infected mosquito feeds on a human, it injects parasites, known as sporozoites, into the blood. These parasites travel to the liver where they develop into merozoites. These are released into the blood where they invade red blood cells and multiply many times. They then burst out, destroying the red blood cells, leading to the symptoms of malaria, such as anemia and fever. Inside some red blood cells, the parasite develops into male and female gametocytes. These are taken up by another mosquito when it feeds on the blood. The gametocytes mature in the mosquito gut and then fuse together to produce a new parasite. This develops into thousands of sporozoites that are injected into a human when the mosquito feeds, and so the cycle starts again. Malaria affects millions of people around the world. Today, malaria infections are restricted to some of the world's poorest countries in tropical regions such as Southern Asia, Africa, and South America. However, if you go back just 100 years, malaria was found in most parts of the world, including Europe and the United States of America.
So malaria used to be a big problem in Europe, all the way up into England. It used to be a problem in the United States. Washington, D.C. was a terribly malarious area, and even up into New England. New York, Massachusetts, there was a lot of malaria. It's now completely gone, so we can eliminate malaria.
A simple way to reduce the transmission of malaria is with the use of insecticide-treated bed nets. These act as a barrier and protect people from mosquito bites at night as they sleep. Nets can be supplied to communities and training can be given to make sure that people know how to use them properly. However, distribution can be a problem and many people still get infected with malaria. There are over 200 million cases of malaria reported every year. The main symptoms of the disease are a high fever and anemia, but severe cases can lead to life-threatening complications. Over 780,000 lives are lost each year because of malaria. Children under the age of 5 are most at risk, and on average a child dies of malaria every 30 seconds. The disease can also cause extreme complications in pregnant women, leading to the death of 200,000 newborn babies each year.
So pregnant women can get massive malaria infections, specifically in their placenta, and that causes reduced oxygen and nutrient uptake to the developing fetus, which can result in premature birth, so you get babies delivered early with a low birth weight, or even stillbirth and abortion can be caused by these placental malaria infections.
Despite the potential severity of malaria, it can be easily treated with drugs that kill the parasite. The use of simple blood tests enable the early detection and diagnosis of the disease. Anti-malarial drugs can then be given to shorten the duration of the disease and prevent further complications and death. A continual challenge to treating malaria is the emergence of drug-resistant parasites. This happened with the drug chloroquine, developed in the 1940s.
In the case of chloroquine, historically we knew that resistance had first come up in Southeast Asia, right on the Thailand-Cambodian border in the late 1950s. And right around the same time, it was reported in the Brazilian Amazon. And then once we figured out what was actually the genetic mutation that caused that resistance, we were able to determine that, in fact, those had been independent events. But the form that came up in Southeast Asia then spread throughout the rest of Asia, and by 1978 it had arrived in Africa and then spread over the next 10 or 15 years through Africa.
In the 1990s, a new drug called artemisinin was developed, but within 10 years, parasites resistant to this drug emerged in Southeast Asia. To minimize the chances of resistance developing in the parasite, malaria drugs are now used in combinations rather than as single drug treatments.
So there's a lot of anxiety in the malaria world right now about our current first-line class of drugs called the artemisinins, because a sort of a mild form of resistance has emerged in the last few years. And looking what happened with chloroquine, looking at what happened with the antifoliate drugs, the fear is that it's going to also spread from Southeast Asia, eventually to Africa. And right now, there's really nothing in the drug development pipeline that would be able to replace it. So if we lose the artemisinin, we're in big trouble.
By looking at the different stages of the parasite's life cycle, it is hoped that targets for new drugs and vaccines can be identified. Genome research is at the forefront of these investigations.
A genome, when it comes down to it, is a list of all the genes that are present in an organism. So the human genome gives us the list of everything that it takes to make a human. In the same way, a parasite genome sequence gives us a list of all the genes it takes to make a parasite. So to a malaria researcher, that genome sequence is a list of targets. It's a list of things that we could develop drugs against, a list of things that we could develop vaccines against. And if you have many hundreds of different genomes, you could see which of those genes are variable, which of those genes are conserved, which of them are essential, which of them some parasites have and some parasites don't. All of that information is hugely beneficial to our efforts to develop new drugs or vaccines against malaria.
There is lots of research taking place around the world to improve our understanding of the biology of malaria. This research is looking at the parasite, the mosquito vector, and the human host. It is hoped that this will lead to new interventions for the prevention, treatment, and ultimately the eradication of malaria, saving millions of lives worldwide.